Feel free to ask questions about any of the topics that we covered today, or even, previous days as well. Or drop off.
I have a question.
Go ahead, Ellie.
Okay. Well, we have been starting with RNA-seq analysis in Entameba. And for following a galaxy and following a flowchart, do you have any pattern that we could follow? Because by now, bioinformatics has analyzed the data for us, and it has been difficult to do it. I haven't tried Galaxy, but I don't know if there is a basic flowchart that we can use so we can start putting our own data. So this could be easier for us.
Yeah, so I think Suzanne was going to, and maybe she did already, or it'll show up later today or tomorrow. She's going to add a couple of exercises that we have from previous workshops, specifically for running differential expression analysis with the RNA-seq data, with your own RNA-seq data. And so those would be good to look through. And the workflows that we put together are available on our Galaxy homepage. They're right in the central portion, and you can pick ones that apply to your data. So for example, let's say you had, Entamoeba, you're comparing, I don't know, Entamoeba trophozoites to something else, or treated with drug or untreated. You could, use the same exercise, just divide up your groups of samples, and then run the workflow that we have on the main page, and see what the results look like. You can also pick from the dropdown. I don't know which Entamoeba species you were working on, but you can pick from the dropdown one of the Entamoeba species that we have in AmoebaDB, and run your analysis like that. So I'd recommend you start there and start with once Suzanne has the exercise connected, you can start there and try it out. And if you still have questions, just click on the Contact Us link and let us know that you need more help. And I'm sure one of us will be happy to either, respond by email if it's straightforward, or we can hop on a Zoom and look at it together and walk you through it as well.
Okay. And the other question, if you have, we have tried transient transfections over expressing proteins, but it's really difficult to have stable transfectants that are more reliable for the information you can obtain. But we tried with transient transfection, and the variability between the groups was very big. So I don't know, in Galaxy, you can deal with those variability to discard groups that are not that are the most different from our duplicates, from our triplicates. I don't know if I get understood.
You're usually better not to do that, actually, because if they're variable, you need to capture that variance in order to do your statistical test effectively, because part of what it measures is the variance. If you think it's variable because something's actually gone wrong somewhere, like maybe you've mislabeled a tube, then that is a good reason to get rid of it. But if it's just variable, then you should keep it, because
in this case the variability is coming from the actual technique, right? Because they're assuming that they're going to have some cells that lost the episomal transcript maybe or something, and then they're no longer expressing it, and so in that case, it's more like a technical issue as opposed to real variability. Is that correct, Catherine, or no? Or what do you think?
Yeah, I mean, I think that, but it's still real variability in that you're always going to see it when you do this kind of experiment.
Yeah.
And you need to take that into account in order to establish whether what you're seeing is real or not.
Okay.
But if you wanted to remove, let's say you wanted to remove that variability, how would you do that? Would you just run the analysis and look at which samples fall out of the cluster, the PCA, for example, and just eliminate those? Or how would you do it?
Well, yeah, I guess. And there are ways you can do it by looking at like the overall variance and how far things stick out. The problem with PCA is because the axes aren't always consistent. especially if you plot them in Galaxy and it just goes with the default axis. So it's very easy for things to look like they're a long way away when they're actually very close to each other.
Okay.
So you want to be a bit cautious doing that. On the axis on the PCA, it'll always say how much of the overall variance that principal component accounts for. So if the whole axis only represents 1% of the variance, but they're at opposite ends of the axis, they're still actually quite similar to each other, if that makes sense.
Okay.
So maybe we can discuss that a little bit more tomorrow when we look at the
Mhm.
results, the output results from.
Yeah.
Okay, thank you.
Is it possible to ask a question regarding the search strategies?
Oh,
Yes.
sure.
Of course.
Yes, perhaps I'll come back to tomorrow when we are looking at orthology. But one of the issues I found looking into Vectorbase is looking, for example, for cytochromes. And as you know, or everybody knows, they're very notorious in terms of nomenclature, in terms of how they're characterized in families, clans, and so on. In any event, just by looking, using the keyword P450 or cytochrome, it returns additional things like glutathione transferases, carboxylesterases, transport proteins, and so on. So one needs to further filter the results to get the P450, the bona fide P450. So that's one of the questions that I have, and I might come back tomorrow with a more specific example.
I'm happy to, I don't know if Gloria wanted to show this, but I'm happy to show you maybe some things that could help you.
Yeah. Go ahead, Omar. I think it's a general question regardless of Vectorbase or not.
Okay, I mean, you could also you could supplement if I'm missing something, but so I'm in Vectorbase. I just did the P450 search as you were speaking, and of course you get a large number of genes.
We're not seeing
So we don't-
Vectorbase.
Oh, I'm not. Am I sharing my screen?
You're showing a screen, but we just see the background desktop.
A beautiful landscape, though.
Yes, yes, that's my other desktop. Here we go. So hopefully now you see the Vectorbase.
Yeah.
Okay.
22,000.
Yep. And then if I select this, the genes, you'll notice that the results were found under these different parts of the gene page, I guess. So you can limit this to the product description, for example, and apply. So now you're down to 6,000. You can obviously explore these and see how they distribute amongst the different organisms in the database. So you see that the numbers are now getting less and less. And you'll see here now, because I only limited to where the P450 was present in the product descriptions. So now you'll start seeing that the names of these genes are becoming a little bit better, right?
Yeah.
And then of course you can limit also based on, I don't know if you were interested in
George, are you interested in a particular species or?
Yeah, I'm looking at basically the Culicidae, the Diptera.
Aedes, Culex?
Basically Aedes and Anopheles and Culex,
okay.
Cuingifasciatus.
So you can obviously certainly go in and limit here already. So you can go in and say, well, I want to only pick 80s or pick all the ones you want, or you can further limit them later on in your strategy. So when you export these, you can use the filtering options that will should show up in a second here on the left-hand side. That's another way of doing it. But also, you may not be satisfied only with running a search for P450. You might then decide, okay, I want to combine with this other types of searches, right? So for example, you may want to limit these as well by, I don't know if the P450s have any sort of interpro domain. I'm assuming they do have something.
They do, yes.
Yeah, so let's do this, and let's pick Interpro domain.
Your search is just loading, Omar. So we are just looking at your search strategies loading.
Okay, let's give it a second.
And it appears frozen now. The wheel is
Yeah,
not.
yeah. It's not okay. Now we've done the experiment. It's not my pointer. So let me do this.
I beg to differ.
What happened? You're gonna stick.
Sometimes it takes a long time to load. Actually, I've noticed that. Yeah.
Yeah, no, it's already on my side. It already loaded and was done. But it's let's see. Hopefully you can see it now. Loading. No, it still says loading. The screen is loading though. Share screen.
I thought the Wi-Fi and a pen would be good. but-
Yeah, it's really bizarre. I'm not sure what's going on here. Let's see. Now I have a feeling it's something on my, because I did have a system update yesterday, so I have a feeling it's something there. So now you can see, I'm just going,
Yes, now we can see it, but we didn't see what you did before that. So
yeah,
maybe
no,
you
I'll
want-
come back. I just want to do a quick speed, so that looks fine. So, okay, so now you should see this. So I exported, right, into a strategy. So I have all my results. I did not filter because I figured I want to keep everything here. So now I can add a step and just look for Interpro. Hopefully that's still showing you what I'm doing.
Yes, Interpro.
Perfect. Okay, I'm gonna select all. Gonna select Interpro from the list. You can select any of these parts, subdomains of Interpro, but I usually go with Interpro. And then we should go with Cytochrome. So I see there's one for site P450, so we can pick this, but I don't know, maybe some of the other ones may be of interest. So you can do an intersect, right, with this, and so you're basically asking anything that had the annotation of P450 in the product description, and also had the P450 domain, Interpro domain in the gene as well. The site is very slow. This is slower than usual. I know we had some bugs yesterday that were affecting the site, so so hopefully, we'll report this to the systems team.
Okay, the next question. I don't want to waste your time, but
No, it's not a waste for sure. So
let's say that I would like to combine this in a strategy, and the notorious things about these cytochromes is that they're involved in all sorts of biochemical pathways, anabolic and catabolic. Catabolic pathways mainly refer to detoxification. The question is, if I use it in combination with the keyword detoxification, to what extent the annotation related to detoxification comes from validated data, literature data, or experiments?
So Gloria can correct me if I'm wrong, but the answer is very little is coming from validated literature and data in terms of that. If I had to guess.
Yeah,
All right, okay.
I will think it's better to do a search query with pathways, KEGG, or something else directly. I don't know if you want to demo that, Omar.
Yeah, so you can, for example, take this list of genes and ask, well, which pathways may they be involved in?
All right. Okay. Yeah.
So you can add a step, and so there are a couple of ways of doing this. One way is you can transform to metabolic pathways. It's not a great. It works okay, but the problem is that this is dependent on EC numbers, and so you can choose whether you want KEGG or Metapsych. So let's go ahead with KEGG. Yeah, so any of the genes, I think that's fine. Exact EC match, let's keep it at that. And we can come back and modify this if it's giving us too many results, but let's run this. So basically it's going to look at the any EC number associated with the genes in this list that we have, and it's going to return the metabolic pathway that enzyme commission number that enzyme may be involved in. Of course, there are going to be some pathways that are non-existent in in your organism of choice, because it's even if a single enzyme is present in a pathway, it's going to return the whole pathway. It's going to say like
Right,
what
right. Yeah.
photosynthesis is there because that
Oh, yeah.
particular so you have to be careful with that and see how many enzymes are actually in your list. I'll give this a few seconds, see what happens. I don't know if somebody who's not demoing Gloria or somebody else, maybe you want to post on the systems channel in our Slack and let them know that the site is very slow.
Yeah, I'm doing it right now.
Oh, thank you. So anyway, when this runs, what will happen is you'll get a step right here, which will list the metabolic pathways. And then actually there'll be a column for to look at the pathway decorated with the enzymes that were in your list, right? And so you can take a look at them, or you can just So that would be the way I would look at that.
Okay. Yeah.
Hopefully this will pop up and we can demo this. If not, we can demo it tomorrow as well. So
You can also do pathway enrichment if that's helpful.
oh, good idea.
Yeah, I've done that. The thing is that the unfortunate thing with this group of enzymes is that the annotations are very hairy.
Yeah.
They leave a lot to be desired. That's the problem. Annotations.
Yeah.
And George, for these P450s, I don't know, Judy Willis from UGA University of Georgia, she manually annotated many of them, I believe in Anopheles gambiae. So that will be the only species for which we have manually curated, now that I remember. I know you asked this earlier, but sorry.
Yeah.
Slightly catch up with my memory.
Thank you. Thanks a lot.
So here's the result. So it showed that there are 59 pathways that were returned, right? And now you can look at them. You can see how many. Let's see. Do we have that unique gene count? Yeah, you get the enzyme count, but then you could look at the decorated pathway. So I don't know if any of these is something that you're. Let's see.
As you can see, there is fatty acid degradation, of course, drug metabolism.
So let's look at the drug metabolism, for example. So when you do that, it's going to, again, depending on how fast the site is, so it's going to load this pathway. And I think Catherine is the one who worked on all of this code. So it's good to have her on because she's the expert on this. But
at least to know it's not just this that's slow, it's everything.
Yeah. It looks like the whole sites have gone slow for some reason. What will happen is this should load a pathway right here where nodes that are highlighted in orange are ones.
So nodes that are highlighted in orange, the pathways come from sources like KEGG, which are obviously not vector specific, they're generic. Sometimes they have enzymes in them that aren't actually represented in our species. So the nodes that are orange, that have an orange highlight, are ones where we have at least one gene with the same EC number. If they don't have an orange border, then it means that we do not that we don't have any genes in the database you're looking at that we know map to that enzyme, which of course doesn't mean it's not necessarily not there, especially if the annotation's poor, but it gives you a sense for what we know is there.
Yeah, that's an excellent, very helpful point, because a lot of these enzymes, they've got in insects, for example, they've got homologs in humans, and there are annotations in KEGG about an enzyme which has the same number, but it comes from the human annotation.
Yeah, and it's always a problem with any sort of functional analysis, whether it's gene ontology term analysis or pathways or whatever it is that often the the ontology or the resource has been developed primarily for model organisms, and as soon as you're not a model organism anymore, particularly you tend to miss lineage specific things because
Yeah.
they're just not there.
Yeah, I'm kind of going on a tangent and looking at Drosophila homologs, orthologs, and see if there are better annotations there. The problem is that Drosophila has fewer genes. These genes, they're duplicates. You know, they've got so many synonymous genes that it's amazing. So anyway, but thank you very much for all this, and I hope I
And
have not taken
George,
too much of your time.
No, one last thing is that because Vectorbase has Drosophila loaded, maybe that's the way you may want to start the query that Omar was just demonstrating for you. So instead of selecting right away your mosquito of interest, you may want to start with Drosophila and transform later your results to your species, to your mosquito.
That's a good point. Thank you very much. Yes, I'll try to do that today.
So the pathways took a while to load, but I thought it was broken, but it actually loaded. It's just that there's a very slow connection. So you'll notice there are two types of colors here. Orange, meaning that at least one EC number was found in Vectorbase that maps to this enzyme, and then purple are the ones that were found in your results, basically in your set of results.
Right.
So, you know, and then you can zoom in. You know, if you zoom in, you can zoom in a bit more, and you'll see the actual reactants, compounds, and reactants, but also you'll see here the actual enzyme itself. And then you can click on these, it'll tell you the enzyme, and then you can explore them further and see all the genes that match this EC number, basically. So
great. Thanks a lot.
Sure. Yeah.
Hi guys. Thank you so much for such a beautiful workshop. But I have a pretty simple and basic question. I'm new to all this sequencing stuff. So my question was, when I get my sequencing results, let's say whole genome sequencing. Now I need to map it with a reference genome. Now, how do I make sure if I go to NCBI and look at the reference genome data set, say for Plasmodium also, I get a lot of information, like a lot of different data sets. Now, how do I make sure which one is the most accurate data set that I'm supposed to select as my reference genome?
So if you're working on falciparum, or
yeah.
So if you're working on falciparum, the most accurate one will be the one we have in PlasmoDB.
Yeah, that was my follow-up question. Like, can I just go to PlasmoDB and download the whole genome as for my reference? How can I do that?
Yeah, absolutely. So let's go back here. Go to PlasmoDB. So first of all, if you wanted to do this in our Galaxy instance, the Plasmodium falciparum 3D7, for example, is already in our Galaxy. So you can already, you don't have to download the reference. You can just run it in Galaxy if you have a straightforward experiment. If you're trying to do this on your own or in your own environment, and you need the reference, then you can go to our if you go under data, and go to where are we? Data, and then you can say download data files right here. And then once you select this, we have this really nice filtering mechanism, and so you can filter on the latest release if you want to make sure you're getting that, and then you can filter. Let's say you want 3D7, for example, and so actually select only these. So selecting only 3D7. And now these are the files that are available for you. So down here is the table that's coming out. And now you'll look at this and you'll see this is, Plasmodium 3D7. There's a GFF file. There's the, protein file. There's a transcript file. Depending on what you need, then you can pick the files. The GFF will contain the annotation and curation, and then sequence files will be available here. If you want to you know,
And the whole genome one's a bit further down. It's the
Oh,
one
there it is.
called _genome. Yeah, that one there.
Yeah.
Yeah.
So thank you. Yeah, good point. So you can sort these actually by file format. That's probably the best way to do it, because you would want a GFF, and then you would also need FASTA file of your sequence. And so now they're sorted. So now you see your genome file right here and your GFF file right here. So then you can just, you know, select these and download.
And you can also filter further in the boxes above that as well, if you want. So
Point.
you
Yeah.
can go to the category, for example, and go to annotation and curation or sequence or whatever, and find what you want that way, if you know.
And if you want to annotate your empty sequence, then you can go to Companion, which our 3D7 is also present in Companion.
If you're assembling a new sequence, if that's what you're doing, but if you're doing RNA-seq, then obviously that you won't need that. But yeah, okay.
Okay. All right, that was helpful. Thank you.
Sure. Yeah. And once again, for everybody, again, if you forget this or you have additional questions, just click on the contact us, and it's, you know, really easy way for you to let you know, a bunch of us on this call will get the email and somebody will answer you. All right.
Yep. Looks
I'm
like
ready
there's
for
a
that.
one-to-one, yeah, one-to-one relationship between attendees and teachers. All right, we'll see you guys tomorrow.
Okay, bye everybody.
Bye, everybody.
Bye, cheers. Good night. Bye.
Bye.